is our Tyrosinase Kinetics presentation by I and Esme, Dixie State University. So, tyrosinase is mostly used in melanin production pathway and can be obtained in various organisms such as bacteria, fungi, plants, and other animals. So, this is an enzyme that it helps speeds up reaction by hydroxylation of monophenols to all diphenols. This enzyme called tyrosinase is very important in enzymatic browning. For example, when you slice up an apple and you leave it out for 5 minutes and it turns brown, yeah, that's the enzyme that is in charge of turning fruits brown. Um, Tyrosinase is a key enzyme that is copper filled with two type 3 copper atoms in the active site. And that is widely distributed across so many organisms. And it is very important in producing melanin pigments, also known as melanogenesis. It has two important catalytic properties. Um, First, it converts monooxygenase phenols into orthoquinones by oxytyrosinase, sorry. And it also oxidizes diphenols to form diquinones as substrates when molecular oxygen is present. So molecular oxygen is the oxygen that is found in the atmosphere. It is a diatomic oxygen.
So with the substrate of L-DOPA, it is an isozyme of tyrosinase. It's a precursor to dopamine, and it is involved in the tyrosinase pathway, and it's used as a treatment in Parkinson's disorder. It helps to decrease the neurotransmitter DA, and that frees up the nerve terminals to help with the symptoms of Parkinson's. So when tyrosinase is inactivated, it can lead to the elimination of copper ions, which are in the active sites, and that in turn deactivates oxytyrosinase. So the inhibitor that we were assigned is L-mymosine, which is a toxic amino acid. It is found in plants, but it's most commonly created in large quantities from legumes. In the literature that I had read, I saw that L-mymosine, when it's given to animals experimentally, they experience hair loss and weight loss, as well as cataracts. And it's also found to block cell proliferation via gene cascades. So it is also, and with that blocking cell proliferation, basically they have been looking into seeing whether or not it would be helpful in cancer treatments, specifically for prostate cancer. So L-mymosine is known to be a competitive inhibitor. It is a slow competitive inhibitor, and it competes with L-DOPA for the active site in tyrosinase.
So as we all know, tyrosinase has four enzymes that are resolved by gel electrophoresis in the polyacromide. Alpha tyrosinase is mainly composed of several amino acids, while the beta, gamma, and the delta tyrosinase are composed of almost the same amino acids. SDS, called also known as sodium dodecyl sulfate, denatures proteins and breaks down cells for DNA extraction. And this same method can be used to activate tyrosinase in different isoforms. And this can be done when there is a lag period, when a lag period is present, and before the attainment of a steady state. Uh, so Agaricus bisporus, also known as the white mouth mushroom, is the main source of tyrosinase. Um, yeah, so that's the organism that we used in this experiment, and we also used there are different types of inhibitors, and multiple inhibitors bind to active sites and prevent translation of tyrosinase to occur. And this is done by controlling their activities, and it can help in the treatment of hypopigmentation disorders and also prolong the browning process in fruit. So basically, the purpose of the study is to examine the rates of the enzymatic reaction that is catalyzed by tyrosinase, and to also determine if the methods that we use, such as dialysis, SDS, and column chromatography, were actually efficient in extracting the enzyme tyrosinase.
So these are our results. So the data collection that we did and how we obtained the data is by performing a serial dilution. And serial dilutions are commonly used as an enzyme assay to determine the approximate concentration of an enzyme by having the most potent and then gradually decreasing in um, a measured concentration to determine the Km, the Vmax, and I think also the Ki, the inhibitory constant.
Okay. So, figure one, this is a graph that shows our enzyme kinetics without the inhibitor. And the R squared values showed everything was more than 0.5, but they were less than 1. Yeah.
And here are our enzyme kinetics with the inhibitor. Our inhibitor was L-mymosine. So as you can see from our R-squared values, these values are not significant because they all fall below 0.5. So this, what we are thinking, looking at this data, is that L-mymosine was not an effective inhibitor of tyrosinase in this experiment.
So our table one shows the catechol concentration, which we calculated from our Km. So it was a serial dilution, and our calculated Km was 4, and then we divided it by half, and then that was how we got it. And this is the relative activity of tyrosinase without the L-mimosine and also with the L-mimosine.
So this is our Eadie-Hofsey plot, both with and without the inhibitor. So based off of just looking at this graph, we determined that it looks like L-mymosine is a mixed inhibitor. The lines don't intersect. The Vmax are not close, neither are the Km. So when we looked at the literature, though, it says that L-mymosine is actually a competitive inhibitor. So here is the equation for our line without the inhibitor, which is the blue line. It's y is equal to negative 0.108x plus 6.5505. Our R-squared value is 0.661, so that is higher than 0.5, so the data did fit the line somewhat. For the line with, for the experiment with the L-mymosine, which is the red line, our equation was negative 0.1095x plus 17.536. Our R-squared value is 0.7066. So it is higher than 0.5. So our data was fitting the line somewhat, and the data is significant. So the published Ki value, or the inhibitory constant, is 0.13 micromolar. And these values are simulated values. They were given to us. So this is just an estimation of how L-mymosine would react.
So this table shows the Vmax over Km, and the Km values of tyrosinase with and without the inhibitor L-mimosine. So the tyrosinase reaction of the catechol with and without L-mimosine was at a pH of 7.0, And just as you said before, the concentration of the L-mimosine is 10 micromolar, and then the Ki value, and the Ki value is 0.13 micromolar. While the calculated Ki value based off our data and what we actually calculated is 0.16 micromolar. Um, and I just want to talk about so the Vmax over Km also shows why we said the L-mimosine is not really an active inhibitor, or we thought it was a mixed inhibitor because our values are like so far apart. And also the Km values, the difference is 0.0013 to zero. So it showed very little to no effect at all in our enzyme.
So when L-DOPA is the substrate for tyrosinase, the Km is 0.2 to 0.87 millimolars. So based off of this kinetics experiment that we did, we revealed that the Km values decreased as the concentration of the inhibitor was also decreased. So the published Ki value, the inhibitory constant for L-mimosine was 0.13 micromolar. And the value that we calculated off of our data is for the Ki is 0.16. So L-mimosine is a competitive inhibitor. It will bind to the active site only in the presence, only in the absence of L-DOPA reversibly. So the fact that we calculated a higher Ki based off of our data means that L-DOPA was preferentially bound. It was bound loosely to the tyrosinase rather than the L-mimosine. And our values do not correlate with the literature. The Km that we calculated was negative 1.09 millimolar, and the published literature was 0.2 to 0.87, so those are significantly different. So some things we were thinking that could contribute to that discrepancy in the data would be bad data that was given to us, or because of things such as experimental errors, like error in the spectrometer, with the reading of the spectrometer, repetitive like freezing and thawing of the enzyme. It could cause misfolding or denaturation in the tyrosinase, or a human error based on us miscalculating the concentrations that we needed to add experimentally that would skew our data. So what I read on some graduate forums to mitigate having a negative Km, because we don't necessarily want that in this case, we could use things such as a non-linear regression or a direct linear plot. So I guess this is common sometimes when converting the taking the line Weaver-Burke plot, because it's the negative inverse. So having a negative does happen, but putting your data into this direct linear plot can help to get a better Ki and get better enzymatic activity from that. Since the difference in our Km between the kinetics experiment with the L-mimosine and then without, since it was so small, we deemed it insignificant. When using these experimental values for the inhibitor rather than the simulated values, this would likely mitigate the discrepancy in our data. So actually performing the experiment would have probably helped our data rather than just being given simulated values.
So here are references, and this is the end of our presentation. Thank you very much. Applause.